thank you for coming to the presentation today. I guess I should say I'm a software engineer at heart, but I'm in the biomathematics program at UCLA, and I build software for statistical genetic analysis. And as part of my dissertation, one of the analyses that I facilitated, coded in Julia, was this algorithm called iterative hard thresholding. For those of you who are familiar with model selection or feature selection stuff, this is it's a signals processing algorithm, and it's got the same spirit as something like Lasso, and I'll expand on it in a bit. And it proves to be very useful. Now, in contrast to other presentations, this will be a little bit heavier on math and a little bit on analysis. But there is I'll have an a Jupyter notebook at the end for those of you who actually want to see how the package works. So to understand why we care, so I'll give you some of the biological background, the context, I should say, a little bit of math, hopefully I don't scare anyone, and then the cool part will be these numerical experiments where you actually demonstrate the worth of this algorithm. So GWAS, if you'd seen this on the announcement for the talk, it means genome-wide association studies. There are these particular genetic epidemiological tools that people use, even today, to analyze the associations or the variances of a disease phenotype in a population with respect to the genome. So you want to figure out, for example, say you have diabetes, you have a population of people, so you measure their insulin levels, and then you genotype. You basically take a sample of each of the genomes, so one for each patient. And then you want to associate the variance in their phenotype with certain regions of the genome. And the way that people do this most often is to use these genotype arrays. They actually sample single nucleotide polymorphisms. They're point mutations on the genome. So here's an example of one of these genotype chips from Affymetrix. And what you do is the genotype chip's not that big. It's about that big. You have some DNA. Say you take it from a cheek swab or blood or wherever you extract your DNA. You put it on the chip. And then you stick the chip into a very large and expensive machine. And what this chip does is that it has a bunch of markers. There are a bunch of oligonucleotides. And it can tell you the value, so a 0, 1, 2 value in some sense, of these SNPs. Now, 0, 1, 2 is particular to this particular kind of study. What you have to know is that humans, those are the test subjects that we care about, they're diploid organisms. So they have 2 copies of every autosome. And so if you have a mutation, with respect to a reference allele, alleles are these sort of mutation, I guess you call it the way you refer to these mutations. You can have a mutation on no chromosome, on one chromosome, or on both, hence the 0, 1, 2. So you acquire your SNP dosages from this machine, and then it didn't show up very well, I apologize, but if you have, for example, a case control study, people with diabetes, people with no diabetes, then you can begin to analyze the differences in these SNPs. Say one SNP is really, really prevalent in one population versus the other. And so what that does is that it tells people, namely the geneticists and the epidemiologists, who want to do further study, who want to do biological validations, it tells them basically where to look in the genome. The genome is still pretty big. It's about 3 billion base pairs. So if you can narrow it to about, say, 100 million, you're already making good progress. So how does the GWAS itself work? Well, so here I'll give you a traditional protocol. Let's say you sample P SNPs on N people, and you have to have some kind of measurable phenotype. I gave diabetes, but you can also have height. You can have any sort of other disease that you care about. Cancer is a really popular one. So then what you're going to do is you're basically going to take every SNP and perform a univariate regression, right? Very simple. You're going to do each of these regressions independently. So let's say you have 500,000 SNPs on your genotype chip. You have 500,000 lines with respect to the phenotype. Once you have those lines, you can construct an odds ratio of that slope, the slope of this line, having a significantly distinct non-zero slope, right? And this is a precursor to obtaining a p-value, right? Because 2 times the log of that odds ratio gives you a chi-squared distribution with 1 degree of freedom. And so you can just look up the p-value on one of these chi-squared tables. From there, you can plot all the p-values on what's called a Manhattan plot. So on the bottom you see the chromosomes. 1 is on the left. You go up to 22 on the right. And then on the y-axis here, you have the minus log 10 p-value. So basically it's giving you the magnitude of, or how small the p-value is. And those of you who have taken basic statistics know that in general, the smaller the better, right? The smaller, the more likely you're going to get published, et cetera, et cetera. So this is a neat way to visualize all these p-values in tandem. So now what you will observe is that some of the regions have these big spikes. And what you should think is that these spikes are fairly significant. It doesn't show up here on the screen, but they're actually horizontal lines. So remember, p-value, so when you do these p-value comparisons, you have to establish a significance level for your multiple comparisons. And this figure actually has a few of them. So there's a line right around 5 times 10 to the minus 8 that you don't see. And what happens is that anything that pops up above that line is considered statistically significant as an association, and that's what you choose for further study. So you have to remember that maybe this is not the best way to analyze the data. We personally take issue with this, those of us who work with machine learning tools, right? So let's go through some of the issues with this framework. The univariate tests themselves don't have great power. So you're actually not really doing a great analysis by taking every SNP individually. There are many reasons why. I won't get into a lot of them because they can get really nitpicky, and they don't really illuminate a whole lot of what I'm going to talk about. The obvious one right here is that the data sizes can get pretty unwieldy. So here I'm saying about 10,000 people and 1 million SNPs as considered routine. I've heard of people do 200,000 people and 7 million SNPs. And so, I mean, that's not, big data people here talk about terabyte and exabyte scale. So for them, it's probably pretty routine. But I mean, it's still really hard to work with a data set like that. Of course, the big problem here is that even if you have, 1 million SNPs on your genotype chip, usually you might only get 4 or 5 that are actually relevant for the phenotype that you're analyzing. So you have to do some kind of feature selection to pull out the ones that really matter. Remember, biologists want to validate, so we can't tell them to look in 1 million spots. We need to tell them to look in 4. OK, lastly, even if you can get those 4 SNPs, as I'm using an example, usually effect sizes, or I guess the regression coefficients, can be pretty small. And so if you have pretty noisy data and your effect sizes are small, it's possible that they might fall into a noise floor. They're difficult to recover. So this is very relevant if you use shrinkage operators for doing your feature selection. I'm going to talk about that in a bit. All right, so we think about this in more mathematical terms. So obviously, I'll use some more mathy things like penalized multivariate regression, right? That's how we're going to deal with this whole univariate test. We're going to actually test all SNPs in tandem. Of course, we need to exploit sparsity. We need to make our computations tractable, right? And so the way we're going to do this is we're going to use IHT, iterative hard thresholding, which is an approximation to this L0 penalized linear regression. Now, those of you who have done penalized regression before, you should just kind of frown in general at this L0 stuff, right? Everyone tries to do L0 sparse regression. People prefer to use a different regression scheme, but I think that you'll like this one. So one thing before we go on, do remember why we're doing this, right? So these biological validation studies that I keep talking about for the genesis is that you have some I used to call him a minion, an undergrad or a graduate student or postdoc, who's sitting there and has to basically fiddle with a bunch of DNA. They have to try to express genes and everything. So you're asking for it was really hard work, actually. We have the easy part. So you want to give your geneticists a very confident answer for where to look when you are analyzing a phenotype, right? Time, money, and insight, right? This is why we do this. So keep this in mind. This is probably the take-home message already, if you haven't noticed. So remember that we're doing linear regression, right? Here I'm showing you a 2-dimensional plot. But if you want to do it in multiple dimensions, it looks sort of like this. It's basically an optimization problem. What you see there is a residual sum of squares loss. So our vector y is our phenotype. It's our response. We have our data x, and you have your coefficient vector beta. Now, if you're in a traditional statistical setting in which you have many more samples than you do predictors, then the linear model here is a very well-known solution, and we're done. That's not the situation that you have in GWAS. So you have to basically reduce that matrix, that X matrix. You have to pull out the part that really matters. So the way that you do this, you have to regularize this particular regression problem, or we call this by adding a penalty. Usually it's a norm penalty, a norm penalty of form lambda times whatever the norm you happen to choose. So by far, lasso is the most popular way to do this. Lasso is taking the L1 penalty and adding it to that optimization problem. That's what you see up here. Visually speaking, what you're doing is you're taking, instead of trying to explain your phenotypes, y, with this particular predicted response x times beta, the lasso will allow you to predict the very same y with a subset of the columns of x and the corresponding coefficients beta. Now, this is well known. This lasso's been around for, I guess, about 20 years. There are all sorts of ways to analyze this. There's no analytic solution for it, but you can get really fast routines to do this. Now, so I want you to notice that I put lasso in a black arrow. It's kind of like my black box, right? So there are other black boxes that you can use. I'll show you two of them. One is called MCP, stands for the minimax concave penalty. It looks actually formidable, but I want you to notice, do I have a, bingo, OK. I want you to look at this part. OK, so what happens here is that this penalty has two parameters. And what you say is that if your coefficient b sub I is less than these particular parameters, then it actually is going to shrink based on the value of beta. And if it exceeds the parameters, it won't shrink. So what you should think about here is that if the value is very small, it's going to try to drive it to 0. But if it isn't small, small here being measured by however you choose gamma and lambda, then it won't shrink it. So this actually can preserve SNPs at a certain degree, the SNP effect sizes. It's very important. Of course, there's also this L0 stuff. This is what it looks like literally. Here I'm abusing the notation. L0 is not a true norm, but this is apparently something that's done very commonly. These actually are typically a little better than lasso, providing sparse solutions. Unfortunately, they're both non-convex. MCP has the advantage that you can make a convex relaxation of it and fit it with coordinate descent. Typically, it's pretty fast. L0, of course, that you have to do subset selection if you actually want to solve that problem. And that's an NP-hard problem. So it's computationally difficult. With this algorithm I'm going to show you, you can do an approximation of it that ends up actually being better than both MCP and lasso. OK, so I talked about sparsity. I'm going to give you a visual of what sparsity looks like in my mind. So this is the lasso optimization problem in a geometric sense. What you're going to see here is these are the level curves around a parabolic object. So imagine it's sticking out at you. And if you were just to solve the linear regression directly, where beta is, that's the minimum of the paraboloid. OK? What you do in a lasso penalized regression is that you care about the intersection of these level curves with this L1 norm ball, which happens to be a rhombus, not a ball. And what you'll notice is that the intersection occurs here. And this is two-dimensional, so we can very clearly see that the beta, the best beta, is actually shifted over here. So it's driving one of these parameters towards 0. MCP doesn't actually have a norm ball, I don't think. So I'm going to give you what it could look like. This is a tetracuspid. It's a particular non-convex penalty. And what you notice is that the intersection here is basically on that axis. Right? So it's actually driven one of the components of this two-dimensional beta straight to 0. And finally, here on the right, what you observe is that it forces one of the components to be 0 directly. So you can see that as you go towards this L0 norm, you're actually enforcing stricter and stricter sparsity. This is something very important to our optimization scheme. If I can give you one example why you shouldn't use lasso in this case, this is it. Remember, lasso is called a shrinkage operator. What do we mean by shrinkage? So observe here. Here's our two-dimensional axis. Say we have this line. It's an identity line of y equals x. And you apply a shrinkage operator, the L1 shrinkage operator. So you solve that optimization problem. And what happens? Well, you're going to shrink towards 0. That's what shrinkage means. You're kind of moving your graph towards 0. So you see that this is towards 0, and this is towards 0. And then there's this zone in the middle, which is exactly 0. That's what lasso does, right? It's driven all the small components towards 0. Here I'm showing you this is actually MCP, unlike the previous slide. MCP, right, is also a shrinkage operator. But you'll notice that it's not shrinking the large effects. Right? That was what you observed before mathematically. But as soon as it gets small, it'll mimic what it's doing in lasso. So it's kind of attenuating the shrinkage from lasso. This is the L0 norm. Its shrinkage operator is known as a hard thresholding, hence the name iterative hard thresholding. And what you'll observe is that it doesn't shrink at all, except in this zone, right? This particular tunable zone. And remember that, of course, I drew this with zone 1. So the lambda shrinkage parameter here is 1. That means that this is basically just clipping your signal. And you can clip wherever you'd like. So if you are careful about this, you can actually preserve the small SNP effect sizes, right? In fact, much better than lasso or MCP. So this is what we do. Iterative hard thresholding is our solution. It's basically doing gradient descent. Here, the shape is turned upside down for visualization purposes. This is a gradient descent algorithm, and it uses this L0 penalty. Has some perks. As formulated, it's actually stable, which is unusual for non-convex algorithms. So it's stable, and it can give you local convergence guarantees, not global. If you want global, you have to do subset selection. And the big perk, no shrinkage. This is important for GWAS, right? You don't want to shrink those small snip effects to 0. You don't want to lose them. So what does it look visually? Look here. Don't look at the math yet. IHT is actually pretty intuitive. So what you'll notice is that we have a point y. And let's say we want to apply one iteration of IHT. What you're going to do is you're going to step in the negative gradient direction of novel f of y with magnitude mu. That's your step size. You have this intermediate point. Now here I'm showing you a two-dimensional figure. So you have three options. You can have 0 non-zeros, which is the origin. You can have 1 non-zero, or you can have 2 non-zeros. Now 2 non-zeros obviously is just the point itself. 1 non-zero, well, you basically preserve the larger the effect size is. So here this is obviously the larger dimension. You preserve it, you send the other one to 0. And that's reflected in the mathematics. So if you have your estimate of the model, what you're going to do is you're going to take your previous estimate. You're going to step in the negative gradient direction. That's the negative gradient of the residual sum of squares with magnitude mu. And then you're going to apply the hard threshold. And this is simply a projection onto a set that has at most m non-zeros. Right? It's actually pretty simple. So what do we do with this? I mean, is it actually helpful? Is it actually useful? We did 3 experiments to show that this matters. The first one here is a simulation. So we have to demonstrate that it actually has better model selection properties than both LASSO and MCP. The second one is a speed test. So this is actually where Julia comes in these latter two. The speed test is that when you do GWAS studies, because they take in so many genotypes, so the dimension of the data matrix is pretty big, but also because you can encode your data in 2-bit integers, there's an advantage to having wrappers that can use 2-bit data, right? It's 0, 1, 2, or missing for those who are statisticians. There is a missing data component. And as opposed to, you can't use any linear algebra routines on your compressed data, so you have to wrap them. And that's what we did. And what we want to know is, do we suffer anything from using the compressed data itself? Now, there's a lot of GWAS data that's already compressed, so there's great value in having the wrappers. Lastly, we want to know, does it actually work on a whole-scale GWAS? All right, so let's start with the first one. This is quite a mouthful, but this is just to be honest. The simulation is this. You simulate some phenotype, so it's all just basically noise. The phenotype is modeled as x beta plus your noise. Here I've given the noise as the same magnitude as the coefficients. We're testing 3 model sizes here in M true. So there they are. Yeah, 100, 200, 300. We have the data is actually from a true GWAS. We're going to use just floating point data for the sake of comparison. So there's no GPU, no Plink, nothing on the side, right? Very simple. Oh, and we're going to cross-validate. We're going to do lots of cross-validations. We're going to try to get some kind of average behavior of IHT. We're going to do this actually for IHT, LASSO, and MCP. All right? So here are the results. What you'll see is model size 1, 2, 300. Here are the 3 algorithms. And we want to know what we want to know here is that if per algorithm, is it actually recovering what is the true or underlying truth, the simulated truth? So in this case, I'll just show you 100 because 200, 300 are all the same story. Model size 100. Here we are with IHT. There are 100 SNPs, there are 100 causal SNPs, 100 true non-zero components in this model. And IHT gets about 94 of them, right? But remember, we're actually cross-validating. So we're cross-validating for ideal model size. We want to know what is, so because we don't know in reality what the underlying model is, right? In a practical setting, you can't just say, oh, it is 10 SNPs, and I know because I know. So you cross-validate for the best model size. And in fact, IHT says, oh, well, the best model size has about 95 SNPs. So this is actually really nice. It's very parsimonious. Even if it's a little less, you've lost some information. OK, it's forgivable. LASSO gets more of the true model size. It gets 96. But it pulls in a lot of garbage, right? Remember, 100 SNPs in this model. And it's giving you 158. So you don't know, in a practical setting, which of those 158 SNPs are actually the true model. So it's obscuring the biological underpinning of your disease. Lastly is MCP. So MCP actually pulls in far fewer true positives. It actually recovers less of the true model. But what it recovers is golden, right? It's very precise in that regard. One comment about, I don't want you to pay too much attention to these compute times. So this may change when we actually get this to publication, but we used GLMnet and we used NCVreg, which are both R packages. So these compute times aren't really that helpful because this is coded in Julia, and you shouldn't be comparing these two. It's apples and oranges. But for what it's worth, the compute time seems somewhat reasonable, right? So it's not like it's taking an excessive amount of time to use IHT. And what you'll observe if you're looking at this very carefully is that the story ends up being the same for 2 to 300. So IHT actually gets more of the truth than MCP, and it gets less garbage than Lasso. All right? So at this stage, we declare victory. We say IHT actually is doing a better job. So part 2, speed tests. So we're going to use the chromosome 1 data, the same chromosome 1 data that we used for the simulation. But it's actually, now we have 3 variants, right? We have our floating point representation, which is what you could use for any of the model selection algorithms. We're going to use the compressed data using this genotype compression that's really important in statistical genetics. And we're going to use it 2 ways, right? You're going to use just CPUs. We're also going to appeal to GPUs. And you'll see why when you see the table. One point, we're actually using double precision arithmetic. So any of you who have used GPUs before, you might wonder why. GPUs are great with single precision. It involves that step parameter mu. I suffered underflow once, and that's all it took to just ask for double precision registers. This actually diminishes performance a bit. GPUs are really good at single precision. They're not quite as good at double precision. So if you keep this in mind, right, you say there is still some room for improvement, potentially. Anyways, the compute setup is 1 hex-core CPU, 1 GPU. So in theory, you could do this on a desktop workstation. Right? You don't need appeal to cluster computing for this particular simulation. So what do we observe? Well, if you do a oh, by the way, I forgot to tell you, this is actually running over a series of models, right? There are about 25 of them in this particular run. So they're all running over the same number of models, but they take different speeds, right? So if you just use floating point arithmetic, that's about as fast as it gets. It computes the entire path, the regularization path, in 10 seconds. If you use the compressed data, you incur a very large time penalty because you're constantly trying to decompress and to standardize the matrix for proper model selection. And the penalty is pretty severe. Here you can see it's about 44-fold slowdown. Now, this continuous decompression and standardization is actually really amenable to parallelization on a GPU. The GPU can do this all on the fly, and it can spread out that entire matrix across the GPU. So if you instead wrap this particular operation on the GPU, then you can rectify the compute time somewhat. It's only about a 5-fold slowdown. So IHT has better model selection properties. And with our Plink wrappers, we can actually do IHT really fast if you have your GPU available. So last experiment. Does it actually work on a real GWAS? So here's the setup. Now we're going to use the whole GWAS. It's 5,000 patients and 330,000 SNPs, plus or minus 3 or 4. There are some non-genetic covariates that have to go in. There are 4 lipid phenotypes. These are actual, right? These are measured on real people. These, of course, are very relevant for people who study cardiovascular disease. Again, double precision arithmetic, same compute environment. And we're going to do 5-fold cross-validation over 100 potential model sizes. Right? We don't know what the true model is, so we're going to guess that it's somewhere in there. And this is what we get. So we can compute for these 4 phenotypes the best set of predictors. They're all pretty small, you'll observe. And we don't take that long, right? So it's about an hour and a half each time. To give you some context about those compute times, I'm familiar with the person who actually did this original analysis. And I asked her, I was telling her about this at a conference, and I said, oh yeah, this is 4 of 9 phenotypes in that data set. I took those 4, I ran my analysis, and it took about 6 hours to compute the best model size. And she says, wow, that really sucks. Because apparently, this woman, when she had done the analysis, had used that old protocol that I told you about, and it took her for all 9 phenotypes 3 months of compute time. So I think this is a pretty good improvement, right? It doesn't take you a month and a half to do all this. It takes you 6 hours. You can run overnight. So it actually, I'll close with this particular point here of this slide. I was arguing that IHT has much better model selection properties. So if you go in the literature, you can actually start to observe any of these associations that it's highlighted. You can sort of confirm because people find a lot of these associations in different populations. So if they're significant, they pop up a lot. So we did some validation stuff, and I'll kind of breeze through it because it's not really enlightening in this context. These are all the SNPs that it's picking up. And you can see that it's actually picking up a lot of truth. There are some quasi-new ones that if you can actually go in the literature, look at them, analyze them, and what we conclude is actually that those are the false positives. That's garbage. It's not real. They're not real associations. But you notice, of course, that there's very little garbage. Look, almost everything is what you'd expect, right? So that's a good sign. Lastly, the last point here is because we're cross-cutting over entire sets of models, you can actually draw these MSE curves. So I gave you these numbers. Best model size is 10, for example, over here. And what you'll observe is that when you plot all these, is that, oh, 10 and 4 are actually really close to each other in mean squared error. So instead of considering just what is the best model size, what are the best model sizes you can get here? So there are a lot of models that are actually give you almost the same predictive accuracy. So you can go back and study and which SNPs come in, which appear here. This is very useful as a diagnostic tool for GWAS researchers. So we're quite happy about that. We enable this, and you don't get this in the original setup of drawing a million regression lines. OK. So we, again, declare victory. We think we're quite happy with these results. Some future things to think about. Can you actually include or exclude or force particular SNPs in the model? So yeah, it's not in the package right now, but you could. More interesting, can you do logistic regression? Or in general, can you do some kind of nonlinear loss? So IHT right now is only defined for least squares. However, there's some research that will attempt to solve this particular optimization problem with an IHT, with a hard thresholding algorithm. There is actually code in my IHT repository that does this for you. It's a little bit buggy, but it's pretty close. Lastly, for those of you who are more statistically inclined, we're doing estimation, but the question is, can you do inference? Can you compute confidence intervals? And in this setting, no. There's no theory about computing confidence intervals. And in fact, this is where Lasso is superior because there is some research in the last 2 or 3 years that'll actually compute, it's called post-selective inference, right? They'll actually compute confidence intervals based on the SNPs in this case that you've chosen. Lastly, it doesn't seem like we'll be able to do these categorical or ordered responses. So we're going to have to do basically continuous responses or possibly binary ones. These are too hard. So in summary, we've implemented this algorithm. We have this tool. It's in Julia. It's fast. It uses all your compute power, so you can run it on a laptop that you should. But it can use all the cores in this laptop. It can use the GPU on board. So it's really flexible in that regard. This algorithm, we believe, is much better than than Lasso and MCP. Has better feature selection, and in a statistical sense, is very good at pruning out the garbage and just keeping what you actually want. Lastly, don't forget that this is important. We care about this, right? Money and time. Money and time. When you write your grants to NSF, tell them money and time. We're going to save you both, OK? Because it actually does matter for the biologists who have to go back and reanalyze and study these associations that you're pointing them to. So I'd like to thank, I have a few people to thank. I'd like to thank my advisor, Ken. Gary Chen is a GPU programmer. He helped with a lot of the OpenCL kernels. Hua Zhou, even though he may not admit it, actually helps a lot here. You'll see him talk in a bit. And then I was funded by NSF, NIH, and then by the endowment of the late Carol Newton. She was a professor, one of the founding members of our department at UCLA. Unfortunately passed away recently, and had actually donated her life savings to the department just to send students to exotic conferences. And that has brought me here. It sent my friends to Istanbul, to Cancun, But I'm really thankful. I'm still thankful for the opportunity to come here, and Carol Newton enabled it. So with that, I'd like to close.